We're here in Chicago at the American Society of Clinical Oncology, where we've been discussing a variety of new papers, new studies, new information relating to prostate cancer in its various forms, and in particular advanced prostate cancer, castrate-resistant disease, and so on. I'm joined by leading experts in the field, Cora Sternberg from Rome, Peter Wysocki from Warsaw, Kurt Muller from Berlin, and myself, Noel Clark, from Manchester in the UK. What we're going to do is discuss a number of studies which have come up, which we think are important, and we're going to go through some of the pros and cons associated with the data. One of the key studies of this meeting is what's known as the CHAARTED study. This is Southwestern Oncology E3805, which is testing the hypothesis that docetaxel given in association with primary androgen deprivation therapy is beneficial. And I'd like to start with you, Peter, just to take us through the findings of the CHAARTED study. Give us a view on how important you think that is.
Yeah, this is really a pretty impressive study. I mean, it involved around 800 patients who were randomized to androgen deprivation therapy alone or androgen deprivation therapy combined with docetaxel. These were chemo-naive patients, and they were hormonal therapy-naive patients. And the results demonstrate that combination of hormonal therapy with chemotherapy significantly improves all the endpoints like overall survival, time to clinical progression, and the differences between arms are striking. In the general population, the difference between both arms, it's 14 months. So the patients treated with androgen deprivation therapy alone survived 44 months, and the patients in the hormonal arm survived 58 months.
So this is a big result. And Cora, can I come to you? Do you think this is a game changer?
This is a game changer. It's really practice changing. This was done by several U.S. collaborative groups. It wasn't a company-sponsored trial. And what's really interesting is it's not exactly all of the patients had this big overall survival difference. The patients who had the overall survival difference are the ones who had a high level of disease, who had bulky disease according to the SWOG criteria of number of bony sites. And those are the patients who had a remarkable difference and remarkable gain in overall survival. The patients who had less disease, at a certain point they modified the protocol, they amended it and allowed for patients with less bulky disease, and those patients, they did not reach an overall survival difference. They're still waiting for even longer follow-up for those patients. But in those patients with a lot of important disease, there's a big difference. And this is very different from the French study that had been reported about 2 years ago at ESMO, where they had patients with a lower quantity of disease. So again, these are patients with metastatic cancer, and as mentioned, they didn't have, they've never had hormones, and these are usually by the urologist treated with hormones alone, but patients who have a lot of disease burden, who have an important improvement in overall survival. So I think this is really practice changing.
It's true to say there are 3 trials in this area, big trials. The GETUG 15, which is a smaller trial which had a hint of a difference but didn't quite show it, and that's going to be reanalyzed in light of the CHAARTED study. The CHAARTED, which, as you very correctly point out, has a heavier bone metastatic load, at least 4 metastases is the figure that was put on it. And the other study, which we'll report probably in spring of 2015, will be the STAMPEDE study, which may well consolidate it. It's probably too early to speculate on that. But as you say, a potential game changer. How will this affect Germany, Kurt? Will Germany move, or will it wait for a consolidation study?
No, I think the data are really convincing, and you can always argue, I want one more study and one more study and one more study. So I think this is not necessary. However, it takes some time until the community really adapts to new data. So what I don't expect really that it will change practice immediately. It will take some time until, especially, I mean, urologists in Germany were a little bit in a different situation, but as Cora has pointed out, in other countries you would need to change the order how the patients are treated. All of a sudden now the oncologists will treat first-line patients. And how that will sort out, I have no clue actually. Germany will not change the game so much because they are all treated by the urologists anyway. But I would expect it will take some time.
And as you rightly say, there are many countries where this will mean a complete change in how things are done. In my country, in the UK, certainly a number of patients who wouldn't be considered for chemotherapy will now be considered, and we'll have to work out which patients are going to get the drugs, which aren't. The road testing phenomenon has to take place. In Poland, how will you manage that, Peter?
Well, we'll have to create much closer collaboration with the urologists, because as yet is the case that the urologists are treating with hormonal therapy, and once the castration-resistant prostate cancer develops, they send the patients to oncologists. And we have to work in very close teams to define for which patient, which kind of therapy will be suitable. So I think once the urologists are aware that for several percent of patients with extensive disease, aggressive chemohormonal treatment is the proper choice, they will send the patients to oncologists right away.
Or you can argue that we need to have more multidisciplinary interactions and work together closer earlier on, not just be sent to patients as oncologists later on in the game.
It's interesting, and I've had a personal interest in following this, that prostate cancer is gradually following the route that breast cancer took some time ago. And you as an oncologist who covers the breadth of a number of cancers, not necessarily breast cancer, you must have seen this, Cora, in work which you've done in the past, that the breast cancer fraternity has a multidisciplinary approach to managing the disease, which is largely led by medical oncologists, isn't it true?
Breast cancer, when you think about breast cancer surgery and breast-sparing surgery that was pioneered by Umberto Veronese, he was working very closely with Gianni Bonadonna when that was done. I was working at Sloan Kettering back then, and we read this. We said, these Italians are crazy. We couldn't understand what was going on. But that was already a collaboration. And the breast cancer physicians, the surgeons and the oncologists, with that collaboration, other collaborations, have been collaborating for a long time. So I think this is is what's happening now in all of the other disciplines as well.
So just finally coming to the issue with this paper about road testing, Kurt, I mean, in a trial like CHAARTED, it's largely good performance patients who get the drug. And you and I know that we've got lots of patients who are really not very fit, performance status 2, elderly, and so on. How are you going to discriminate? Have you got any thoughts about this?
Well, that's probably too early to think about that, but as you know, as always happens, there will be some arbitrary decision that some patients are not eligible for that primary combo. Like if, as you say, if they are too old, and also again, you know, to have a differentiation between what is a high tumor load, what is not a high tumor load, what is aggressive disease, what's not. That's one thing. On the other hand, keep in mind, here we're talking about metastatic patients, primary metastatic patients, and they're, despite all the discussion on early detection and so forth, they're getting rare and rare. So it's not a, you know, this is not probably a huge crowd out there that where we need to take the decision and say, is that really a candidate for the combination, or will that particular patient be better off with hormones only?
So what I'd like to do next is move on to this, or expand on this issue of road testing new drugs, because, Corey, the abstract which has been looking at the open access program for abiraterone acetate in patients with metastatic castrate-resistant prostate cancer is presented at this meeting. And this is an open access program which follows on from the Cougar 301 trial, where patients had specific criteria for entry to the trial. Could you just take us through the key findings?
Well, this is an extended access protocol with exactly the same criteria for entry as the Cougar 301 trial. So we're talking about patients with metastatic castration-resistant prostate cancer who have failed hormonal therapy, and who have failed 1 or 2 lines of chemotherapy, one of which had to be docetaxel chemotherapy. So this is exactly the same population of patients that was treated in the Cougar trial, in the Cougar 301 trial, which led to the approval of abiraterone acetate after chemotherapy. That trial had 1,095 patients. This trial was started to allow access to other countries and many other physicians who didn't participate in that trial. And we had over 2,000 patients actually in this trial with the same entry criteria as the Cougar trial, but they weren't all of the same physicians or investigators who had been in the Cougar trial. In fact, we had patients from 23 different countries, but it was Europe, it was Latin America, countries that didn't even participate in the Cougar trial. And the interesting thing is that by using the same trial, the same entry criteria, and giving this drug to a more widespread general population of investigators who had not participated in the trial, many investigators who had not participated in trials, was given to urologists and to medical oncologists, what they found in particular was that it was a safe drug. The safety was very similar to what was seen in the Cougar 301 trial, and the efficacy data were very similar. So it can be considered in a real-world population, this drug could be given. It was given until it takes a long time for regulatory procedures after a drug has been approved to actually have this approved and reimbursed. So, for example, in my country, after 301, the FDA came to our place to inspect us in 2011. It wasn't until 2013 that the drug was actually reimbursed in Italy. So we had a lot of access through this extended access trial, and this provided a way for many other places and other countries where it took time for the drug to be either approved or reimbursed.
And it's true to say the access in different countries has been very patchy, largely according to the GDP of individual countries.
Exactly.
I know in Poland you have had difficulty accessing right at the start. Peter, just tell us some of the difficulties that you might have had with this kind of a prescription of drugs of this nature.
Well, actually, all the innovative drugs are pretty expensive, and therefore Poland, as a HTA country, every decision on reimbursement is based on pharmacoeconomic analysis. And the negotiation between the company and Ministry of Health took a lot of time till the drug became reimbursed, and the drug became reimbursed in January, so this year. So it took us like more than 1 year to get the drug for our patients.
But is it reimbursed before chemotherapy as
No,
well now?
only
No,
after.
the same in Italy. It took 2 years for after chemotherapy, and in America, the day after they came to our place, it was on sale the next day, and it's still not reimbursed pre-chemotherapy.
So most of us struggle one way or another to get it, except in Germany, where it's freely available.
Yeah, they throw it out of the window. Huh? But we're in a comfortable situation. That's right. It's you can get it in pre-chemo situation.
But you have a kind of a slush fund that helps you out too.
We do.
Even though you're supposedly in a bad situation.
This is what's known as the Cancer Drugs Fund, which is an amount of money which the government has made available for developmental drugs until they're approved by the UK regulatory authority, which is the National Institute of Clinical Excellence, or NICE. And there's a process that one has to go through in order to get those drugs. Having registered with the Cancer Drugs Fund, it's relatively straightforward, but it's true to say at the moment, in the pre-chemotherapy space, abiraterone is consuming probably about 60% of the National Cancer Drugs Fund. That's right across the cancer board. So, as you said, Peter, these are not cheap drugs, and they do bring with them problems of economics. Kurt, I want to come to you with 2 other abstracts, because this follows on with this notion of new drugs, development of just how good they are, because we have 2 abstracts. One which is looking at the primary, secondary and quality of life endpoints from PREVAIL, and the second is what was thought to be a very promising new agent, orteronol, TAC 700. The results of the phase 3 trial have been somewhat disappointing. Just take me through the PREVAIL data first.
Yeah, it was patients in the pre-chemo situation, mildly symptomatic, asymptomatic, huge trial, close to 700 patients, 1,700 patients. It was enzalutamide versus placebo, so no prednisone in the placebo arm, as in the abiraterone trial, and also in the Rotarinel trial, which we will talk about in a minute. And it made all it was co-primary endpoints: overall survival, radiographic progression-free survival, positive for both endpoints, with a hazard ratio of 0.7 for overall survival, and a hazard ratio of 0.2 for progression-free survival, which is quite remarkable. Good PSA response in the range of 80%, means PSA decrease more than 50% for 80% of the patients. And also with all the other secondary endpoint, time to cytotoxic chemotherapy, worsening of quality of life measured by FACT-P, and so forth, the trial was positive. So as I think quite convincing data, and it will lead to the certainly to the approval of enzalutamide in the pre-chemo space. And so we will see, and that's going to be the question of the future, what we'll start with. Will it be enzalutamide or will it be abiraterone?
Well, I'm going to come to that question in a sec,
right,
because
okay.
Clearly an active drug,
Yeah.
But treading right on the toes of another newly arrived active drug. Cora, the sequence question. I mean, in Italy, if this drug is available quickly, which would you give first, and what would influence your decision to give this first?
Reimbursement. First of all, I would have to say reimbursement. But honestly, we have never had a head-to-head trial of one against the other, so we don't really know which one should be given first. What we seem to know, looking back retrospectively in all of our trials, is that patients who have had abiraterone respond less well to enzalutamide afterwards. Patients who have had enzalutamide respond less well to abiraterone afterwards. So there's something about the second drug that doesn't work as well. Mechanisms of resistance are instilled. So I think that the kinds of trials that are going on are looking at combination trials and looking at different sequencing of these agents, kinds of trials that are going on. We know that when you give enzalutamide, for example, you lower the androgen receptor and you raise the level of testosterone. When you give abiraterone, you lower the level of testosterone and you raise androgen receptor. So Eleni Estefaniou, for example, has presented a very interesting abstract in which she's combined the two. She's done bone marrow biopsies. She's trying to figure out, I mean, how we can decide which one of these drugs should be given first or should be given second. Is it dependent on the level of testosterone in the bone marrow biopsy, for example? And we just don't know that. And there are a number of trials that are ongoing to look at that. We can talk about that later if you want.
Well, we may as well talk about it now, because this is an interesting abstract which has combined enzalutamide and abiraterone in the way of Eleni Estefaniou's group.
Yeah.
It's been done in a very thorough way, looking at translational aspects of the research. And that's very helpful in understanding the pathophysiology. Hopefully we will get some information as to whether the combination will be beneficial. Do you think you might consider looking at this, or are you looking at this in some trials in Germany, Kurt?
Well, I think to look at it in a real phase 3 study will be lengthy, complicated, and probably will not happen. That would mean you have to test the combination against the sequence. And I doubt that this will happen anytime soon.
Well, in fact, in the STAMPEDE study at an earlier stage, this month, should I say June 2014, the combination of abiraterone and enzalutamide goes in as the next arm. STAMPEDE has been recruiting at between 100 and 140 patients a month. It took something like 18 months to recruit 1,900 patients to the abiraterone comparison with ADT, combination with ADT. So I think that earlier on in the disease process, this is a question which will be addressed and hopefully answered.
I'm participating in a trial, it's called PLATO, in which we give enzalutamide up front in patients who've had no chemotherapy, and at a certain point, when they progress, they're then randomized to either abiraterone and prednisone and enzalutamide, or abiraterone and prednisone and placebo. So this is sort of looking at the sequencing.
Yeah, but that's not the same question. That's a different question.
Yeah,
That's
it's an add-on.
If you keep the patient on enzalutamide or not, that's what you answer with that.
It's a little different.
Yeah, that's so. But
you know,
having really the question coming up if it's beneficial for the patient to have him on the combo from the beginning. That's probably a question we will not having an answer for. I mean, Stampede again. That's a little bit a different situation in terms of the disease stage. And so, yeah, I mean...
It needs to be evaluated.
Yeah, it would need to be evaluated, but I doubt that's going to be, that's going to be happening anytime soon.
No, we'll have to wait for that, and I think we think we've got a financial headache now.
The financial headache is the problem. I think it could be a financial...
But I think the sequence is the most important problem, because it seems from the mechanistical point of view that enzalutamide should work following abiraterone, because there is still the receptor. And on the other hand, there's a lot of in vitro data showing that enzalutamide leads to activation of several other signaling pathways that activate receptor, or even some downstream pathway below receptor without ligand or without any activation of the receptor by ligand.
It should work, but
it should work. But Cora has mentioned that if you look at the small studies where we got results from, it drops your response rate by half, and really it works. So it's really the first drug you give works best in all these androgen receptor targeted things, and then the next drug you give works much less, and then the third probably it's really you have to think about if you need to change the mode of action. So that's probably the most important information we have right now.
But there is a new abstract, Peter. I want to come to you with this, which looked at produced some very interesting data looking at androgen receptor splice variants, particularly the splice variant V7, which seems to code for resistance to enzalutamide and abiraterone. Would you just talk to us a little bit about the data from that study?
It's a very interesting study because it shows a novel approach towards determining the mechanism of hormonal resistance to hormonal treatment, because we know that the V7s, there are several splice variants of androgen receptor, and the V7 is, I think, probably the most important because it creates androgen receptor that does not possess the ligand binding activity. So therefore, such an androgen receptor is not responding to any hormonal treatment that targets directly the receptor, like enzalutamide or like abiraterone. And in this study, the group decided to analyze in circulating tumor cells the presence of V7 splice variant of androgen receptor, and they have demonstrated that the presence of the V7 androgen receptor splice variant is responsible for lack of activity of abiraterone and enzalutamide. And this is a very interesting approach, showing that we can analyze in patients without the need for tumor biopsy. We just need to isolate circulating tumor cells and evaluate the mechanism of resistance to treatment. In this case, the resistance to hormonal treatment.
So we've been looking for sequencing rules and stopping rules, and this is a rule which potentially would drive us away from starting the treatment. But it's a sophisticated technique. Circulating cells with quantitative RT-PCR, is this for prime time or is this for research time?
It would take, and if that is really confirmed by other groups, that this would be a way to predict how certain patients respond to treatment. So this would be one of the first predictive markers at all for prostate cancer. It will still take some time, and this could be really introduced in mainstream clinic, because as you say, it's a complicated way. You first need to get the tumor cells, and then you need to RT-PCR, to get it analyzed. So and this will, again, we've been talking about reimbursement. It's going to be expensive.
Yeah.
And so this is probably a major hurdle to get that introduced into clinical practice.
The standardization is going to be quite a lot of fun.
Yeah,
And,
but it's a major advance, and we need to understand the mechanisms of resistance to these drugs. We can't just be giving these drugs to thousands of patients and keep doing trials like we've done. So I think this is the kind of thing that we need to start doing.
Absolutely.
And it's an important...
No doubt.
Also
good.
from the economical point of view.
Also.
We've had a lot of success stories with some of these drugs, and the TAC 700 or Teranol story, has been somewhat disappointing. Kurt,
It's right.
you've been closely involved with this.
Yeah, well, we've already earlier had the results of the post-chemo trial. Teranol had a similar program as abiraterone had. And so now we got also, and that was a negative trial, post-chemo, and now we got the pre-chemo data, and I mean, in short, it was also a negative trial. It was about the same setting as with the Cougar 302 trial, again, like 1,500 patients, and it met one of the co-primary endpoints, which was radiographic progression-free survival with a hazard ratio of 0.7, but it didn't meet overall survival, hazard ratio 0.9, and this was, I would say, it was a surprise and was disappointing, and for the time being, I have not a very good explanation how that happened, because the mechanism of action is not the same as with abiraterone, but very similar. So what I would have expected that it would work as well. I mean, we had some problems with abiraterone in the 302 with the overall survival, pre-specified P levels and so forth. But at the end of the day, the hazard ratio in 302 was 0.7 again, and here we have 0.9. So it's not really. And they did also stratified by region and so forth. It is not clear for me at the moment why this failed.
Cora, have you got any thoughts on this?
Well, I have a couple of thoughts. It's possible that this is a C1720 lyase inhibitor, and maybe it's weaker than abiraterone, maybe. But I also think it's not a great idea to be third. So we had the abiraterone trials, we had the AFFIRM trials with enzalutamide, and all three of them, all of these trials were the exact same setup, the same set of patients, exactly the copycat trials, and it's not good to be third on the market or third around. And what happened for sure in the post-chemo was that there was a lot of use of other drugs. The use of abiraterone became on the market, enzalutamide became available, cabazitaxel chemotherapy became available, and patients got a lot of crossover, particularly patients from the placebo arm. So this particularly was a problem in North America. So if you looked at the ELM-5 trial, the post-chemotherapy trial, it was very clear that patients in North America and in Europe got a lot of crossover of different drugs. It's not quite as clear on this pre-chemotherapy trial that they got all of the crossover. It's not as clear why this trial failed, or if it's a weaker drug, or just because the other drugs were available. I'm not quite as sure that the regional thing is as obvious as it was in post-chemotherapy. But there's a lot of, it's kind of what happened to us in renal cancer. We just had so many drugs and people were crossing over and getting one over another, and we couldn't show the overall survival difference any longer.
And this is important because evaluating new drugs in this area of prostate cancer is going to become increasingly difficult.
Yes.
Do you think that, as in renal, we'll have to move to progression-free as the end point? We've tended to stay with, for various reasons, with survival, and progression-free has always been an important secondary endpoint, but not the primary one.
But we don't have an endpoint because the majority of patients with prostate cancer have bone disease, and it's very difficult for us to measure bone disease. We haven't really validated the CTCs as a validated endpoint for progression-free survival, and we don't have the round things that we have for using RECIST criteria. So that's been the difficulty. Overall survival has been our golden endpoint, and we don't have another endpoint.
So we've had a terrific time so far with new data, new information, interpretations, some of it positive, some of it negative, some of it uncertain. But what is for sure is that we will continue and need to continue to carry on thinking about what we do, testing out new agents, and things will change, that is for certain, and we have to adapt to that change. Thanks very much to our experts, and thank you for listening.